For the session is Dr. Pradeepa Madapura, Associate Professor at Queen Mary University of London, has completed his PhD at CSCR, Bengaluru, where he worked on chromatin remodeling during spermatogenesis. Did his postdoctoral fellowship with Professor Wendy Bangmore. During his work, now he is working on understanding how the chromatin proteins, histone modifications, and non-coding RNAs regulate gene expression with relevance to genetic disorders and tumorigenesis. Today, Dr. Pradeepa will be talking about the precision editing PROTACs to investigate mechanism of neurodevelopmental disorders. Over to you, Dr. Pradeepa.
Thank you very much. Thanks to the organizers and the chairs, and thank you all for staying for this talk. I've heard really good thing about CMC Vellore, but never been there. It's the first time. I'm really excited. Yeah, it will be great. It makes sense why people say really good things about the amazing scientists, clinicians here. So my lab focuses on epigenetics and chromatin, how genes are precisely expressed and regulated in different cell types during development and disease. And what happens when there is a perturbation in gene regulation due to these perturbations in epigenetic mechanisms. So I'm going to today this is the broad structure of my talk. I'll be briefly introducing about gene regulation and epigenetic regulators, how they play a role in gene regulation, and how mutations in some of these epigenetic regulators cause neurodevelopmental disorder, and why specifically epigenetic mutations cause neurodevelopmental disorder. Focusing on a protein called BRD4, which is very well known in the cancer field. And we identified recently this mutation. I'm trying to understand, we are trying to understand the disease mechanism with this mutation. And the second part, briefly touch upon preeclampsia. It's a pregnancy-associated hypertension. The reason I want to present this is I realize I think it's a lot of clinician audience and students. I really liked, and I think it's particularly relevant to India and sub-Saharan countries, and there's a lot of maternal complications much higher. So I'd like to touch maybe in a couple of slides for that. Okay. So quick introduction about chromatin gene regulation. So so the genes are expressed very particularly at particular cell types and tissues, and this is mediated by chromatin modifiers and remodelers and histones, for example. They get modified post-translationally. They get acetylated or methylated. This is an example of promoters of active genes having lots of acetylations, and this particular modification, that's histone H3 lysine 4 trimethylation, and acetylation at many lysines on the histone H3 and H4. Same thing happens on the enhancers. They can be far away from promoters, but they still regulate genes by interacting with promoters. These enhancers also have these acetylations. So these acetylations are mediated by some of the enzyme complexes like a P300 CBP and MSL cat8. I'm just giving only a couple of examples. So that's all about activation, gene expression. There are also genes which need to be silenced, or they are silenced in most of the tissues, and they get expressed only when they are needed. So some group of protein called PCG, that's a polycomb group of protein. There are two complexes, polycomb complex 1 and polycomb repressive complex 2. So they basically maintain the repressive environment at some of the genes by depositing a modification called H3K27 trimethylation. That's a lysine 27 gets methylated by polycomb. Sometimes they kind of coexist with some acetylations. It's something very interesting. They retain bivalency. That means they also have repressive mark. They also have activation marks. So that means they are in a situation where they need to be quickly expressed when they get a signaling or differentiation cues, or for any kind of rapid activation. They don't need to change the entire chromatin. They can quickly change their expression status. Right, so that's kind of our view of gene regulation. The reason I'm mentioning few chromatin proteins in this side is because these are the structural proteins or architectural protein. They play a key role in looping enhancers to promoters. Like, for example, they play a key role in loop extrusion. That means basically allows the DNA from one part of the genome to come together to the regulatory elements or the promoters so that they can communicate, and that's really crucial for tissue-specific gene expression. So these are the group of protein that's this is called SMC complex, and the protein which loads this SMC or cohesin complex called NIPBL. Okay. Why I am saying this particular complex is it's very interesting. There's a rare developmental disorder, neurodevelopmental disorder called Cornelia-de-Lange syndrome, which is still one of the very common rare developmental disorder. It's still rare, but it's one of the more common one, maybe just below Down's syndrome. So 1 in 10,000 to 1 in 30,000 people affected. So this is a major kind of phenotype what we see: developmental delay, intellectual disability, microcephaly, and you can see craniofacial abnormalities, kind of raised eyebrows, and these are very common features clinicians would look out and then suspect it could be a Cornelia-de-Lange syndrome or CDLS. So very interestingly, most of the cases, nearly 70% to 60% cases have a mutation in this particular gene called NIPBL. This is a loader of cohesin protein to the chromatin, which plays a key role in transcription and gene regulation. Still, it doesn't really explain why this as you can imagine, this complex must be playing a role in almost every cell types and tissues. Why particular features, craniofacial features and developmental disorders? That even puzzling to understand, because this is another protein, BRD4. It's a very important chromatin protein which is important for reading acetylate histone and known to have various function in the cells. We identified this mutation as well causes the kind of similar phenotype as a CDLS, and we ended up showing that actually they BRD4 indeed interact with this complex, and that explains why they have an overlapping phenotype. So this is the BRD4 in the top, and BRD3 and 2 are the two other genes of the same family. They almost have the same domains. What they do is like they recognize acetylated lysines on the histones through this bromodomain 1 and bromodomain 2, and also bind to acetylated proteins, not non-histone proteins too. So they have been kind of exciting targets for many cancers, because this protein, particularly BRD4, is important for upregulation of MYC and basically MYC mediated cell the transcription. So the cells, or particularly the cancer cells, gets kind of adapted to high level of MYC and high level of proliferation. So that can be targeted by targeting BRD4 so that it reduces the transcription of these oncogenic enhancers or oncogenic super enhancers. So it's an exciting epigenetic therapy in a cancer field. But the problem is these proteins have a very wide variety of function, not just in gene regulation and transcription. They play a key role in in the DNA damage response and splicing, and they are very important processes, and it can have a pleiotropic effects. And also another key thing is why central nervous system particularly is been affected when you have this perturbation of this histone acetylation or BRD4 axis, because we know not just BRD4, there are many chromatin modifying enzymes or the readers of histone acetylation are mutated in neurodevelopmental disorders. So what we do is we use human embryonic stem cells and then we create or generate kind of exact patient mutations in these cells, and then use differentiation system to neuronal progenitors, neurons, or brain organoid, mini brain organoids, and then try to model and see why what are the defects and how we can study them. So the other really interesting approach we use recently is because when you make these genetic mutations in the cell line, and it can have a long-term, because the genetic mutation is a long-term, you grow them on the cell culture system, it can have a pleiotropic effect. So what we need to do is basically try to understand or mix a combination of other approaches which we can test immediate effect of these perturbations. So what we use is a few class of inhibitors. These are inhibitors very specific to bromodomain 1 and 2, and also PROTACs. So these are basically, I'll come in the next slide what are these, but these are basically target these proteins rapidly for degradation within minutes and hours. That's really exciting in the field. I think if anyone who is a chemist would understand, I think it's a growing field. The way Tan Mong was mentioning about these molecules, these small molecules can potentially add it to the cell, and they can take the pathogenic protein for degradation within minutes. They are really exciting. So there are a lot of interest in developing PROTACs for BRD4, because it's we know that when we knock down BRD4, you can kill the cancer cell very quickly. But the problem with some of these inhibitors, including JQ1, is although they are already in clinical trial, they may be a bit effective, but they are kind of failing in clinical trials because of the toxicity. As I said in the beginning, in the earlier slide, that it can affect many cellular functions. So this is the PROTAC which I just mentioned in the last slide. So this is another level of PROTAC. It's a very specific PROTAC in a way. So we can create a gene, like endogenously tagged protein like BRD4 using CRISPR-Cas9. We inserted a tag called FKBP, mutant version. So what it does, like if you have a heterobifunctional molecule, one part of the molecule binds to this mutant FKBP12, other part of the molecule binds to endogenous polyubiquitination pathway, and basically takes this protein for degradation within minutes and hours. So you can see 1 hour of treatment of this molecule in this cell, the BRD4 level almost goes to undetectable. It's really cool, you can very rapid as well, and any consequences you see, whether you do RNA-seq or phenotype-seq, you can kind of mostly say that it's all due to direct effect of BRD4, not due to any indirect consequences. That's a PROTAC, so you don't need to tag. For example, you have a patient, you have a mouse model or something, you can simply feed or inject this PROTAC. You can remove or degrade the protein in minutes and hours. So we use this approach in human ESLs and neuronal differentiation. Quickly go through some of the data what we have. So what we found is a very interesting change. Instead of seeing a downregulation of many genes, we started seeing upregulation of genes within few hours of treating this drug-mediated degradation. So, and most of these upregulated genes appears to be neuronal lineage. As you can see, they are important for axon, they are brain specific, but all this data is from embryonic stem cells, human embryonic stem cells expressing neuronal genes within hours of treatment. In fact, we have used the combination of DTAG and PROTACs. DTAG is basically one which I mentioned in the last slide about direct tagging. We give exactly similar results we get. A lot of genes are upregulated. So we do also a lot of epigenomic methods like a chip-seq or cut and tag or cut and run to profile where these histone modifications and chromatin proteins are. Interestingly, we found BRD4 actually is not just in the active genes, it's also in the polycomb repressed genes. So that's, as you can see, a BRD4 level correlates with the K27 trimethylation. This is surprising because we didn't really expect that. And then we could profile that, and we identified a kind of a new mechanism that we think BRD4 recruits to these acylations in these repressed genes. So what are these genes which are upregulated? They appear to be bivalent, many of them. That means they have both repressive and activation mark, and the BRD4 plays some kind of role to keep these cells in this repressed state to prevent premature expression of these genes in the developmental lineage. And we wanted to understand the mechanism, and when we look at the mass spec data for BRD4 intractome or proxiome, we found very interesting that BRD4 indeed interact with some of the polycomb repressors, PRC1 and 2, as you can see. And then we wanted to move on to see the model, organoid model. So we generated its cerebral brain organoids and then performed single cell RNA-seq and bulk RNA-seq as well. And also we had this organoids with the patient mutations. And we also use combination of PROTACs. So we wanted to quickly check what happens in the organoids when you deplete BRD4 within a day, for example. So we see upregulation of, again, the bivalent polycomb repressed genes. They are very important for memory formation. These are basically immediate early genes. They are immediate responders for any learning and memory. The massive deregulation. When you analyze the single cell RNA-seq data, what we found interestingly is like there's a particular lineage getting prematurely kind of differentiated, and we are having early neurogenesis, particularly towards this diencephalic lineage. These are kind of a forebrain neurons, particularly which gives rise to optic vesicles and retinal pigmental cells. As you can see, these are the mutant. There are a lot more optic vesicles and retinal cells compared to a wild type in the left side. Which you can actually visualize in a brain organoid. This is the wild type. You can already see kind of eye-like structure forming in the brain organoid. Of course, the forebrain gives rise to eyes, so with unexpected we see much earlier time point. When we look at the mouse model, we see exactly same. Again, we see mutation or knockout of BRD4 gives a same vision, eye phenotype, nervous system, and skeleton or neurological phenotypes. That's really assuring that these the organoid models are really useful to model the disease and also test potential therapeutic therapies, including these molecules and PROTACs. So that's a working model. Just what I have said so far is that BRD4 appears to interact with the repressive complex unexpectedly, and then seems to be a new way of maintaining the precise regulation of gene expression in the developmental stage. Particularly the neurodevelopmental lineage is affected. We still don't know why neuronal lineage, but we think it could be lineage which need to be tightly regulated, and that gets specifically perturbed. Okay, and then moving on to the next topic which I wanted to briefly touch upon is on it all started from my lab when we were studying a genome regulation. So genes are only 1% of, or exons 1% of the genome. Nearly, although the around 50% is not really anything it does, but in another 45 to 50% is actually transposable elements which are basically derived from ancient infection of virus in our body. They basically integrated in our genome millions of years ago. So they've been silenced, and they have been the cell as a mechanism to suppress these transposable elements, so they don't cause any kind of perturbations, but some are still expressed. They escape this suppression mechanism, and they can some have a developmental role because the cells also, or we also have co-opted some new function for these transposable element. What we showed basically is like some of these transposable elements which are epigenetically modified, and they can function as a regulatory element. They act as enhancers to regulate genes, right? And some of these transposable elements are really important, again, particularly in trophoblast or in the placenta, for example. Some cell types really need this transposable element genes, couple of genes. They make the outer layer of placental villi, that syncytium layer. So we do need some of these viral derived DNA in our genome, and also we need them for regulating gene expression as well. But however, when we get older, or because of the stress which we accumulate, or due to DNA damage or shortening of telomere, some of these transposable elements get expressed, and that can cause inflammation. Particularly, it can lead to kind of a sterile inflammation, we call it. There is no infection, but it increases the inflammation. That kind of symptom happens also in some situations. For example, aging-associated inflammation and neuroinflammation. What we found is like in preeclampsia, this is about 5 to 10% of the pregnancies are affected. It's basically a sudden increase in the blood pressure in women more than 30 weeks of pregnancy. It can lead to a lot of complications. The only way is like early delivery, or it can be prophylaxis, can be aspirin low dose, but only if it detected early stages. It's quite, as you can see, it's quite a serious issue. We found that while studying some of the placenta from preeclampsia, we found these transposable elements are upregulated in the preeclampsia, and that causes the sterile inflammation in preeclampsia. We went on to kind of develop this into a potential preventive or kind of a diagnostic or a predictive signature by looking at the level of these transposable elements in the pregnancy, particularly if it collect the blood around 30 to 16 weeks of pregnancy when woman goes to first visit, and then we can perform isolate plasma. And so far we have done a RNA sequencing. The hope is to go into a RT-PCR or a loop-based amplification method, so we can quickly predict the chances of someone getting a preeclampsia later on in the pregnancy. So this can be tested before 20 weeks of pregnancy, while the symptoms actually occurs around more than 30 weeks. So it's very early, even before the experience, we can test it. So I just wanted to say that I know that there are a lot of clinicians here. If anyone working on preeclampsia or access to samples, I'm really happy to collaborate with them, particularly in the Indian context. Please feel free to reach out to me, or if you know someone who has access. So with that, I'd like to thank my lab. So most of my this work is done by Fanny and Manthan, or a postdoc in the lab, and some contribution from student Ziba, and also work continued by new postdoc and collaborators. So our funding is from Medical Research Council and BOT Charity. Thank you very much, and happy to take questions.
Thank you, Dr. Pradeepa. Now the session is open for discussion. We will take 1 or 2 questions.
Yeah, wonderful talk, Pradeepa. So you see microcephaly phenotype in BRD4 mutations, but it is not recapitulated in the brain organoids. So do you have any comment on that?
Sorry, what I didn't get the last part.
The mutations in the children, they have microcephaly phenotype, right?
Yeah.
So the brain organoids, do they have the microcephaly phenotype, like a small brain phenotype? I don't see any difference in the brain, the organoid size of the organoid.
Oh, okay, good question. So it depends on how we studied. I think we did have a mutation, homozygous mutation we created first, and we saw really severe organoid microcephaly phenotype. But the thing is, we couldn't really keep, because the organoids stopped failing to make a long-term growth because of the homozygous mutation. The heterozygous mutation, we didn't really see smaller brain phenotype in the organoid.
Is the children homozygous or the heterozygous?
So the one which I showed you, it's a heterozygous mutation.
Okay, thank you.
But we do see in some cases.
Yeah. We will take one more question, please.
Hi, sir. So I just want to ask, in the organoid model, if you're introducing these mutations at the embryonic stem cell stage and deriving into organoids, you would also get that mutations in other cells of the neuroectoderm origin in the organoid, like the astrocytes, oligodendrocytes. Do you see it there also?
Yeah, because this mutation is in the stem cells, so we take the stem cells into organoid. Yeah. But I think at the stage what we are studying, around 63 maximum, we don't see many astrocytes in there. Okay. Most of them are neurons and progenitors.
Okay. So within that stage itself, you do all the studies so that you can differentiate the effects. All right. Okay, thank you.
Hello, can you hear me? So can you explain me in a preeclampsia can you explain the correlation with the preeclampsia?
So the preeclampsia, as I said, it's a pathophysiology is really not well known. So what we think is there's two different mechanisms we are seeing this. One is early onset, so that could be defective placentation, and the late onset we see because of crowding of placenta, a lot of senescence and inflammatory phenotype that causes this.
Most of the resistance happens at the spiral artery with this in-situ interface. So how this is going to really affect means I didn't get that point. Your mechanism. It's not clear to me.
Sorry, I didn't have much time to go through. But basically, what we are taking placenta after delivery, and then we identified a signature, and then went back to 13 week of pregnancy to see whether we can get the signature early on in the blood plasma. So, and the cells, when the syncytium, when they are growing, some of them undergo necrosis, and that cells release to the bloodstream, maternal bloodstream, and when you take a blood from maternal and then isolate plasma, you get some of the signature coming from the syncytial.
I understood that, but how the correlation is made? How you made the correlation with eclampsia? That's what my question means.
Oh, we know the outcome of the pregnancy, so we wait for the outcome of the pregnancy, and then I'm happy to discuss that later.
Yeah, thank you.